So, I apologise if I explain this really badly. I sort of understand it, but I don't know how well I'm going to explain it verbally. So, T. brucei, which is an African trypanosome, can't get into cells to hide from the immune system, so it has these proteins on its surface that helps hide or defend against it. They're called variant surface glycoproteins, or VSGs, and they're sort of like amino acid / antibody. So it has an N-terminus and a C-terminus and an anchor. The anchor is what attaches it to the surface of the trypanosome. And it goes through monoallelic exclusion. So there are 2,500 different VSG genes, and there are 15 expression sites, but only one is expressed at one time. And it's transcribed in a high amount, so there's a lot of them there when it's expressed. This is because it prevents, it's sort of like antibiotic resistance. So if it expressed them all at once, then the immune system would develop and react to all of them, and then it'd be like sort of stuck because its one defence is now recognisable by the immune system. But if it does one, and then the immune system learns what that is and fights against it, and then it changes to the second, the immune system has to relearn it, so it does it like one at a time after each other. And they can do this very fast, so there's a high turnover of VSGs on the surface. It can take up to 12.5 minutes to fully change the surface over to a different VSG. And they're also fluid because of the anchor. So because they're on the surface, they're shielding the cell membrane of the parasite from the immune system and the antibodies present. So the antibodies bind to the VSGs, but because the fluid, as it's doing the spinning motion of movement swimming up the midgut, they're sort of blown off it into the flagella pocket. When they're into the flagella pocket, they can then be broken down, and then there's no antibodies on the surface for the immune system to recognise and come and attack. So here's sort of a diagrammatic representation, if that helps at all. So you can see you have a lot of them with the VSG red, but then that's recognised and they start being attacked by the immune system, so they start to drop. So they switch to the blue one, more blue, they're recognised and attacked, so they drop, switch to yellow, and so forth. On the other hand, T. cruzi, which causes Chagas, can do immune evasion. So they can hide within vacuoles of cells where they replicate. This then causes a lysis of the cells, more are released, more infect other cells, they replicate, and so on and so forth. When they first enter the body through the wound, they first infect macrophages and neutrophils. They then cause acidification within the vacuole, which then leads to lysis of the vacuole. The T. cruzi are then within the cell membrane, the cell cytoplasm, where they can replicate, and then once they've replicated, they cause cell lysis. Also, the secretions of T. cruzi can cause inactivation of B cells, so they no longer, not inactivation, but a feature in B cells to change so that they don't express antibodies to the T. cruzi antigens. They also reduce the number of B cells. But they also affect the T cells because they actually do activate the T cells, but because they don't have the PAMPs on the surface, the triggered T cells can eliminate the full infection.